Yeah.
I'll try not to be here on Friday if possible. No. Here we go. Yeah, I know. I can't. It's very. I can. I believe me, we do plenty of remote lectures. I save them to the end. Yeah, just trying to figure out what place to go on spring break that we can go to. I'm trying. We're trying. I don't know if it's going to work. It's always my wife's mom. She's been there since Friday and won't be back until tomorrow night or Wednesday morning. I'm running over there after classes today. All right, here we are. So this is kind of, you might remember where we left off, everyone. And where we left off, we go back, we looked at the different oxygen levels. And again, being familiar with it, an aerobe or strict aerobe has to have, have oxygen available, but they make catalase and superoxide dismutase. There's, as you can see, they give you a nice list of what's on there. That's part of it. Microaerophiles only do well in deleted amounts of oxygen, and these are important organisms, so they can't be directly exposed. The Helicobacter pylori, that's the famous one associated with sometimes with ulcers or certainly gastric carcinoma. Again, when we talk about it, you're going to see that there's a strain of streptococcus that causes quote unquote flesh-eating disease, which we cover in wounds. And Borrelia burgdorferi is very famous. That's Lyme disease, but it's never really a problem in the sense that it goes from bloodstream in a tick in the animal to the bloodstream in the tick to the bloodstream in us. So it's never really exposed to full levels of oxygen. And you'll get to know Borrelia burgdorferi. Doctors are vain. So the Willie Bergdorf was the doctor in Connecticut, in Lyme, Connecticut, who basically identifies this. Facultative anaerobes, okay, if it's around, they use the oxygen. So many of those gram negatives we work with in the lab. Staff, to some extent, works with it. That way, it's not really a true aerobe, but it will use oxygen. So when they say, again, facultative is an important word. Something is facultative. It has the faculty or the capacity to grow fairly easily under any circumstances. The next ones that we'll get to after this have another great word called fastidious. And you should know the distinction between facultative, which really, again, doesn't require a lot to grow, versus fastidious, which requires something special, whether it's conditions, whether it's nutrition. Okay, organisms that are fastidious have a lot of difficulty growing in general environments. So those are an important distinction. That's there. Obligate anaerobes are just that. I mean, you wouldn't even class them because they can't detoxify oxygen. And what's interesting about aerotolerant is that oxygen isn't a player. They don't have the mechanism, and that's the basis when you get into lab of the catalase test. They can have superoxide dismutase. They don't have catalase. And as you will see, the big distinction between Staphylococcus and Streptococcus is, even though neither one of them is a quote strict aerobe, okay, is the catalase if the catalase test is positive in Staphylococcus because it contains it, versus Streptococcus, which does not. And that's a really significant test in the laboratory. It's the science behind it. So all of those are worth knowing. This is when we start to get into fastidious organisms. CAPNO just means carbon dioxide. So as tough as anything to grow is this Neisseria species. Gonorrhea and meningitis are extraordinarily noteworthy, significant diseases, life-threatening diseases. Some Neisseria meningitis, immediately life-threatening disease. It's bacterial meningitis. Okay, so all of those are important, and they're very, very hard to grow. In the lab, we have a tough time identifying it because on the general augers that you use in the lab wouldn't grow at all. They have to use a special auger called chocolate auger, and they mix that with some other chemicals because if other bacteria get into it, they suppress it. So if you look it up, it's a very unique auger that facilitates the growth of Neisseria species. that is there. So that's really a classic fastidious organism. Brucella, you don't see a lot in this part of the world, and strep pneumonia. And when you start to really grow streptococcal species in the lab, we use blood agar. So again, it derives extra nutrients from the hemoglobin in red blood cells, and in doing so breaks it down. And when it does that, we're able to identify it, whether it totally consumes it or only partially consumes it. So you'll also do those tests in the lab, and when we start looking at streptococcal diseases, particularly strep throat, you'll see how we identify those. So they, again, that even by, so in other words, we often will use what's called enrichment agars for these, because we put something extra in them because of their special needs. For our purposes, really the only ones we have to worry about are bacteria that grow, because really for our purposes, pH 6 to pH 8, we theoretically wouldn't exist. Maybe internally we wouldn't exist. These are oddballs. They have alkaphiles and acidophiles. These are called neutrophiles. I'll write it out for you because it's different. When you get to the blood, for those who haven't had that unit, this is the most common white blood cell is called a neutrophil. It stains neutrally. Bacteria, we add an E, neutrophiles, because they like neutral pH, roughly between 6 and 8. So really all the bacteria that we encounter, and really that's, of all of these, the most important of the pH levels. So, I mean, we have some extreme ones that are really, what happens is, the only one of these that is classically an alkalinophile likes higher pH, okay, to neutralize urine and colonize and infect the urinary system. The there's like 3 big bacteria players for urinary tract infections common. E. coli, because it's fecal contamination, Proteus, because it's fecal contamination, and an oddball called Staphylococcus saprophyticus. And all of those are somewhat noteworthy because urinary tract infections, particularly in women, are extraordinarily common. And normally easy to treat if they're caught early. For those of you in nursing or clinical fields, that's the problem with UTIs is they move fast. There's nothing insignificant about that. So that's not, just drink a bunch of water for a couple of days and come in and see us. No. Let's get a urine sample and see what's going on. Right? Yeah, that's not something, it's not one of those let's just wait and see type of things. So there's certain things that clinically you're much more alert to. And depending on where many of you go, particularly in the nursing, if you end up being nurse practitioners, that's the kind of thing you got to be very, very alert to. We, again, don't deal a lot with these ones that are in highly salted concentrations and things like that. The most interesting one of these that we call a halophile, and you'll do this, like, what's on for this weekend lab?
I don't know.
He's here. I saw him this morning. I will not be here. I'll be in another room. I'm sorry. I'm not going to pop in and say hello at all. So halophile means salt loving. Okay. Normally we see these in salt water, in salt lakes, in those kinds of climates. Most salty environment for bacteria that affect us is your skin, which is a salt plane. So the way we isolate staphylococcal bacteria and identify it as to whether it's Staphylococcus aureus, a nasty pathogen, versus Staphylococcus epidermidis, which is normally not a nasty pathogen, is we grow it on a particular agar. The reason I asked the question, we call those selective and differential agars. When something is selective, it means we've added a chemical that encourages the growth of one or certain bacteria and limits the growth of others. So when we want to grow Staphylococcus, we make salt, we put 7% salt in the agar. Differential means we can see a visible change to help us identify it. So what you'll see we do in the micro lab all the time is you add dyes, typically pH sensitive dyes, because when bacteria flourish, they ferment sugars. So mannitol is the sugar in question. It's called MSA or mannitol salt agar. And we had a very famous chemical dye called phenol red. And when acidity occurs, it changes from red to yellow. Like you all did, you know what phenolphthalein is? Didn't you do that like in high school when all of a sudden you had something that turns pink? You put a drop of a clear liquid in a little test tube and it turns red or pink. Well, it wasn't that long ago. I can remember. I graduated high school in 1969.
Oh, no way.
Yeah, God bless your heart.
I don't think you have the right number.
59. My sister graduated in 63. Oh heavens. Okay, moving on. Don't look at me strange. Don't like it. Don't like it. So you're going to see in the lab you're going to use MSA a lot. You're going to see a red agar and you're going to look for it to turn yellow. I'm not so worried about the radiation aspect of it. It's not something we utilize typically. We will talk more about radiation in the next chapter, which is methods of control. We use a lot of ultraviolet and we use a lot of ionizing radiation. Dr. Dress, I'm curious, and maybe he will we have an ultraviolet lab that we will do here sometimes, depending on what we have available to us in the schedule. So, because we have an ultraviolet in that back prep room in the micro lab, there's an ultraviolet like thing that's in there that you can turn on, and we expose bacteria for certain amounts of time. It's a cool lab. And then pressure, we don't see this with like at the bottom of the ocean. You can imagine what pressure would be. So, by and large, we see a lot of that live in combination habitats, multicellular organisms like us, and there's a variety, and they can be helpful, they can be harmful, they can really have no impact whatsoever. So there's a great deal of complexity that's really not for this course to describe. These are interesting terms that you see. These are the kind of terms that you might notice are test worthy. The significant one to me is this, parasitism. Okay, so I'll give you an idea. So we interact with other individuals all the time. So by and large, we have some degree of symbiotic relationship, meaning we exist together, we have the same food supplies, the same living space. So typically, if you have to be in a dorm, it would be nice if there was mutualism. So if there were two people in a room and you liked your roommate and you got along, that'd be good. That's mutualism. They live in an obligatory, you're assigned there, but mutually beneficial. This one likes to clean, this one likes to be tidy, whatever. Think these are real life analogies. I never could understand the significance between mutualism and commensalism. The partner called the commensal receives benefits while its partner is neither harmed nor benefited. To me, they're the same kind of thing. Nobody's hurt. So to me, mutualism and commensalism are very similar. They are, are they mutually beneficial, or one person it's not as beneficial as the other? Who cares? This is the bad one. This is my daughter's first boyfriend. A parasite.
Oh, okay. I see.
Lived in our house, didn't bring anything to the party because his parents kicked him out. For a while.
How long?
Too long. Until he was arrested. It was.
Oh, okay.
Oh, come on, you had that boyfriend. You're both of you. Your dads probably had the same reaction to your first boyfriend. What? What? Don't know what you're missing.
Any mention of a guy my dad looked at?
My My daughter had an extended bad boy period.
Ooh,
That would be the best way
okay.
to describe it. Okay. It's all right. He's actually not a bad person.
But
Just a bad, look, when you grow up with when your mom is a drug addict, it doesn't help. It doesn't help. So I'm not forgetting. He's got two wonderful brothers who are normal. Go figure. He just never can get himself away from that stuff. So parasitism, and this is really what an infectious bacteria virus does to you. It's a relationship where you give them a place to live and they hurt you in some way, shape, or form. I'm not trying. Forgive me. I'm not trying to cast any aspersions. I know most dads don't like their daughter's first boyfriend.
or any.
Or any would be a good way to express it. My dad was the poster child for that. Whoever my sister picked, it's unacceptable. Okay, and there's other ones where the antagonist. Oh, who cares? That's not. And we'll talk about synergistic relationships. These are general terms that synergism basically, to me, that's like a good relationship where the combination is better. The sum of the parts increases. But this is interesting. Gum disease, cavities, blood infections are where bacteria help each other accomplish bad things together. Cavities would be the best example. You have that entire biofilm, which is a multi-layer complex, and what it's doing is weakening the enamel in your teeth and facilitating an anaerobic streptococcal organism to give you cavities. And this is where they talk a little bit about the biofilm that's there. It starts with one, they all get together. I mean, there's not a lot to say about it. Okay. And interesting that the combination has a basis where it tends to activate or enable certain genes to express themselves. So, I mean, one organism benefits and the other one is harmed. That's a parasite. So now the term parasite is a little confusing in micro. Parasitism is a generic term for this. One is benefited, one is harmed. When we use, if you said, oh, Mrs. Jones was in, we're treating her for a parasitic infection. The implication of a parasitic infection is that it's a eukaryotic pattern. So when you hear the term in a hospital environment, parasitic infection, it generally, because they would specify, it would usually apply not viral, even though you could make an argument, or not bacterial. Typically, the implication of parasitic infection is that it's a eukaryotic pathogen. So that could be anything from burn, fungus, or yeast infection. Protozoan, probably the classic one of these again, malaria. So it's a whole different ballgame that are there. So, and you'll see this repetitively in your studies as you go longer in your careers. There's a whole lot of terms that you really have to clarify specifically. Okay, when you use the term, some context is helpful. Okay, this is the important part of this whole chapter. Boils down. They put it at the end. I'd have put it in the beginning. Okay, this is called the bacterial growth curve. We want to understand how bacteria. It's very important. When we get a bacterial infection, we're a living thing with unlimited space, and for a bacteria, it's an unlimited amount of nutrients. Okay, so they can just keep growing, okay, without any limitation. But you can't study bacteria by infecting somebody and seeing how well or how bad they do to kind of characterize their growth. So where do we do it? We do it in the laboratory. So when you do it, when an infection affects you, okay, We call it an open system. Limited space and limited nutrients. In the lab, when you put bacteria on a plate or in a test tube, it's called a closed system. There are limitations to the amount of space and a limitation to the amount of nutrients. And why is the space important? Waste products. Because it's metabolism. They're gobbling stuff up and there's waste materials that are produced. That's the significant thing. So that's the jumping off point to understand the bacterial growth curve. One cell becomes two. Okay? Binary fission. Bacteria, with very, very few exceptions, this is how they grow. They start with two. You can't even see the parent cell enlarge because they're so small. Chromosomes duplicate. And it's just like you saw that animation with the endospore. It appears to enlarge, boom, separates. Now you got two identical ones. And a septum or a wall forms. So you've seen it 1,000 times. Boom, boom. Technically, the genetic material bonds with the membrane to use a platform temporarily, duplicates the DNA. The wall forms, they divide. Bingo. All done. Nothing to it. We look at the time. It's classically referred to as the doubling time because all it does is double. You start with 1 or you start with 1,000. 1,000 double, then they double again and again. So every generation, if you want to call them that, is a factor of 2. And as long as the environment, this is the big number, remains favorable, like in an open system like us, it will continue. And that's why rapidly growing problematic bacteria, I mean, my microbiology professor said this like repetitively almost every lecture. Staph can kill in 24 hours. It doesn't take long, if not being treated, to be lethal. So the typical, what we call doubling time or generation time for most bacteria, 30 to 60 minutes. For E. coli, it's probably 20 to 30 minutes. We studied that a lot. Okay, some can be even shorter. Some can be very long. Leprosy, days. Okay, tuberculosis, 10, 20 hours, something like that. Maybe a day for it. That's why they're slow going. So it's not, oh my God, someone had tuberculosis and I was in a room with them. Don't go crazy. Okay, it's long sustained exposure. Same thing, that was the misconception biblically when they shunned lepers. I mean, unless you were, when they put them in leper colonies, it only got worse. So, I mean, the misunderstanding, and I can't emphasize this enough. Many of you are going to work around sick people. Understand that what can make them sicker and how it can impact you or other people in the hospital or in the office or wherever it is. It's really important. I mean, that's why we do this stuff, so you can understand when do I have to take a lot of precautions, when is it's no big deal. I just wash my hands, I'm fine. So some in bacteria you can see are odd. Most pathogens, one of the reasons and how they overcome you is they grow awful fast. So there's a lot here. You can look at the chart. I'll do it my way. So arbitrarily. So here we can look at the time in hours. Okay. So if we start with 1 in a matter of 5 generations, that could easily just be 2.5 hours. Now we got 32. If we had started with 100, now we have 3,200, 1,000, 32,000. Okay. So when you draw the curve out, we use a logarithmic graph. So most of you in high school had that graphing calculator, that TI-83. Is it ringing? We didn't have that. Okay, we had slide rules. I know. Does anybody know what a slide rule is? We have a winner. When I was in college, we didn't have calculators. We had a calculator. Your phone does more than that calculator. We chained it to the wall in the chemistry lab so no one would steal it. Your cell phone does a zillion things more than that. But the idea is we could use what was acceptable is using your slide rule. slide rules are based on logarithms, and indeed, logarithmic calculators and TIA3 are based on that. Just to give you a little context. So what it means is this. They use this because this is the number of hours. This is how many grew. If we use this in simple rather than 200, 400,000, all the way up to 1,000,000, we spread off the paper. Couldn't see it. You can see in a matter of less than a day, if we started with one bacteria with a typical doubling time, we would have over 1,000,000 within the course of a day. It doesn't take long. That's the take home. When you plate something out in the lab, in order to be able to see that dot that we call a colony, you have to have a minimum of 1,000,000 organisms. So you can go back there 2 hours. If you played it out in the lab today and went back tonight, if you could get in the lab, you wouldn't see any change. Tomorrow morning, you might see something. It takes a little while for it to grow. So we look at this, okay, the doubling time, and we're able to create. This is the. I used to do a calculation. I got threatening emails from students. You don't want to see that. But effectively, this is it. So if you have 1,000 organisms, and you basically start with a number, so it's times whatever the number of organisms with this, you could say, number of organisms times the number of doubles. So again, in 3 hours for half an hour, that's 6 doubles. So 2 to the 6th power, in that, or 2 to the nth power, I don't know if that's that. 2 to the nth power, so you have 1,000 times 64, because 2 to the 6th power, 2 times 2 squared. You get the idea. That helps. I don't do it because I don't need to do calculations that are here. Hopefully no one will course it. So we figured out how to take this information and extrapolate into a predictable pattern of bacterial in a closed system. Means we can't get rid of the waste products, and we're going to use up the nutrients. So what that means to a bacteria are happy as long as they have plenty of room and enough to eat, and there isn't a bunch of waste products hanging out. So we would inoculate identical cells, identical tubes of broth with a certain number of cells. We would incubate, and at regular intervals, we would take samples out sterilely and count the number of organisms after we plated them out on agar. And so we developed this type of thing. So you could incubate the plates, and after an hour, you really couldn't see anything. It didn't mean they weren't growing. There are just not enough of them to visualize randomly microscopically. After 2 hours, you start to see, and then more, and then more, and then more. So you would be able to see it in the tube, because the tube would get gradually more cloudy. You would see an element of turbidity indicating growth in the tube. Great. So by looking at the data, they were able to construct a chart. So how did the start? It started with, because either it wasn't growing very fast or you couldn't see them, what they call lag phase. Whenever an organism gets put into a new environment, it takes a certain amount of time to adapt to the environment. It takes a certain amount of time to gobble up nutrients and start metabolism. That's the lag phase. So we don't see any tangible growth. Once it's up and ready to double, it doubles extraordinarily rapidly. That's called log phase, which is short for logarithmic, because that curve you saw, that exponential curve, is a logarithmic curve. Okay, pH is logarithmic. That's why the Richter, whatever that scale is, the Richter scale, rather, is the scale for when you talk about earthquakes, logarithmic. So, it's 10 to the power. This is also a kind of logarithmic curve, except it's 2 to the power. So it grows even more. So when you grow the curve, it increases geometrically, and effectively it creates exponentially. And so exponential growth is rapid, and it grows very, very rapidly. Then it plateaus. So as long as it's growing, enough to eat, enough room, not a bunch of waste products, it grows. Once it starts to slow down, it will flatten. We call that the stationary phase. It doesn't mean it stopped growing. It means that as many are growing as are dying. We're measuring the number of viable organisms. So it's not that there aren't ones that are living. Some are living, some are dying, but it's roughly equal. But it indicates the rate is slowing down. Depleted nutrients, depleted oxygen, lots of waste products that are there in the media. Eventually, there's so much gone that cells begin to die at a similar but slower rate, a similar pattern but slower rate than they started to live. And the speed again is dependent. And then even we will see some viable cells that exist. I've tried to culture them after a year or so, having them like in the refrigerator and where the liquid is still there and it doesn't appear to have changed, but you can't get them to grow again. This is what it looks like. That's a very famous curve. And we deduce certain information. It isn't so much the curve, but the ones that are easy to understand is lag phase. It takes a certain amount of time for it to become accustomed to the environment and grow. It's about 5 hours, sure, maybe a little less, maybe a little more, depends on it. Once it starts to grow, and again, this is logarithmic, so that's not 2, 4, 6, 8, that's 10 to the power. 100, 10,000, a million. Get the idea? Logarithmic. We used to use all this. Or the Asian credit. What would we do without it? And we still were able to figure things out. And Einstein guy was around long before any of that. Go figure. And exponential growth. And notice they show you kind of the yucky stuff too. So that exponential phase typically goes on in a typical bacteria for the better part of that first, 10, 30 hours, somewhere in the middle of 20. And then it begins, and you'll notice the right way to draw this is not, it's not like geometry class. It's not a trapezoid. Okay? It's round. So it's not I'm fond of saying living systems don't make sharp turns. So it's like we're growing and then, we're not growing anymore. Doesn't work that. We call that technically, in the old days, we used to call that late log phase. It's a little confusing, but really what is happening, this is when it starts to run out of nutrients and the waste products are accumulating excessively. Eventually the nutrients run out. That's when it goes to stationary phase. Yet, some bacteria are surviving, some are dying approximately equally. Well, if they're surviving, they have to have nutrients. What would they be eating? The dead bacteria. When bacteria die, they explode, and living bacteria gobble them up. Dead. Doesn't eat that anymore. See, I used to do the doctoral lecture. He's a professor. Down to the lab. Lava. Ceranthus. Silence of the lamp, fan to your eye. Help, Lord help me. Stranger. So that brings us to stationary phase. And that continues till we begin to get death phase. And death phase is always, as you can see, not as sharp as living. What's the big deal? Lag phase is accumulating nutrients. Log phase, the big thing, it grows. What's the take home from that? It starts to grow very rapidly. That is the absolute best time to treat an infection. When things are growing rapidly, all they do, is gobble up anything in their environment. So if you add an antibiotic, something that will hopefully kill them, or at the very least slow them down, they will, it's like a ravenous dog. It will eat anything you put in front of it. Unless it's highly trained in bacteria or not. But I'm not trying to hurt the dog. You get the idea. The other thing that it tells us is we get certain things called primary metabolites. Those are useful waste products. So in genetic engineering, when we're making insulin, human growth hormone, hepatitis B vaccine, whatever, okay, that's when you're harvesting. Because we designed that effectively to be a typical secretion or primary waste product from bacteria. So we can keep an open system in a lab. And basically what we do is once we get in that phase, we feed nutrients and we siphon off waste products. So we keep it constantly going. So that's what they do in a laboratory basis. In stationary phase, two other things. One, if it has the ability, and not many do, to form endospores, will. And most importantly, secondary products called metabolites. Doesn't matter what the chemical is, something is produced by metabolism. Secondary metabolites. The ones in particular that we harvest here are antibiotics. Because bacteria don't produce substances to kill or harm other bacteria unless conditions are adverse. It's a defense mechanism. So not all, so again, they don't all produce endospores, the ones that do, because endospores are created when times are adverse. And the same thing with antibiotics. You're getting an adverse living condition, and you're going to basically it's a survival instinct. Keep saying it all the time. Bacteria are living things. They're going to respond like you and I will. We'll fight to survive by whatever means necessary. And that, everybody, is the bacterial curve. So, exhibit phases of rapid growth, slow growth, death. Microbes in exponential phase are more vulnerable to antimicrobials and heat. Treat infections early. Medically, early identification and treatment is the single most important thing. You can't, and you take it home. I mean, sometimes you have to respond aggressively. I'd rather do something. Some things it's okay to do nothing. Okay, somebody has a mild fever. If they get these things recurrently, you may let it go. I mean, you over time you try and get an idea when something abnormal is occurring. Cells in growth phase are more vulnerable to conditions disrupt their metabolism. Actively growing cells are more vulnerable to growth inhibition and destruction. They're just vulnerable because all they're doing is growing. If you can put anything in the environment to affect it, do so. We basically, this is created less by sampling than more by turbidity. We looked at degrees of cloudiness as the measuring. I've been in labs in colleges and universities where we use spectrometry, and we don't do it here, but you could. There it is. So we look for turbidity a lot to create this curve. When you do antibiotic disc testing, we don't do it here, but to set it up, you're supposed to use turbidity because you want to. That's a way of seeing how cloudy the tube is and standardizing it, because then you have an idea what the concentration of microorganisms is. Any test that you do scientifically, you want to try to have as close to identical standards. And all turbidity is how well or how poorly light goes through it. So, I mean, these have been around, and the reason we talk about these have been around for a long time. Yeah. spectrometry. We still do it all the time to enter things. And they do a test to see, oh, we want to see somebody die. We want to see if there are any toxins in their blood. So what you basically are doing is you're running a spectrometry, basically GLC, gas liquid chromatography, and you're doing a series of tests on it, seeing certain patterns, and you try to match the patterns up with one another to see what the substance is. They, on all those crime shows, they start to do that. So many universities do the same thing. Other ways. In the old days, and when they probably developed this curve, they did a lot of what are called direct cell counts. So you'd have like a slide that had a grid, and you'd count it, and you know how big the grid was, and what the volume was, and you do and plug it into a formula. Nowadays, we use Coulter counters. So you put a fine stream of the material in, and it is either measured by light or electrical deflection. Basically how it works, they use genetic probes. That's what I meant by a direct count. This is the way they used to do it in the old. They used to do blood tests that way. When they did a red cell count or a white cell count, you just did it visually. So we didn't have the sophistication and technology. Here's the Coulter counter illustration. That's there, and that's that. So that concludes the material for exam 1. Don't thank me. I will bring you now, therefore, to this. This is what's coming up next. A little introduction. Yes, thank you very much. I have a handout for all this. I have taken the time to outline this for you. Don't thank me. Send money. Preferably send the money.
Tuition.
Hmm?
I pay my tuition.
I understand that. It's all right. I'm joking. My wife says I'm going to get fired anyway. I'm just going to do it like, you can't ask the kids for money. Ask their parents for money. Right. So this is the unit entitled Chapter 9, start of the next test number 2. Here we are. Barely past Groundhog Day. Stupid groundhog. And you can't rely on a rodent to tell you the weather. And there's nothing really, if you've ever been there, there's nothing exciting in Punxsutawney. Not even Gobbler's Knob. You like Punxsy? The downtown is okay. You can't walk from there up to Gobbler's Knob. It's up a giant hill. You from there?
No.
Weird. So, now you're out of the area. I listened to a sports show, and the guys, Mike Tirico, who does the NFL on NBC games, because when he was working for ESPN, says, we were driving from like New York to Cleveland, and he goes, and we saw a sign that said Punxsutawney off of Route 80 when he got over like to Du Bois in that area. He's like, we went to see it. He goes, guys, because they'd just seen the movie with Bill Murray. Not much different. We went there. So this is all about control. Now, there's other methods of control that are chemical. That's antibiotics. Different chapter entirely. This is disinfection and sterilization. It's very much a definitions chapter, and very much the first of our chapters that are very clinically oriented. This is useful in everyday life, and this is particularly useful in a hospital because we talk about sterilization, we talk about disinfection and decontamination and sanitization, antisepsis. They're not equivalent. When you sterilize something, you sterilize something. When you disinfect, when you disinfect. If you use an antiseptic, it's not a form of disinfection. Okay. We have a pecking order of problems, starting with endospores. We start to look at terms like microbicides and microbostatics, bactericides, bacteriostatic, fungicide, fungistatic. We start to look at different targets, not so much in this chapter, more so in antibiotics. It's really important. So this is These are definitional type things. We do not use these. We do not use this one. Okay. So the key player is sterilization. Sterilization is the destruction of all microbicide or microbial life, including bacterial endospores. The caveat to all of this, it does not include prions at this point in time in the definition. We do not know for certain scientifically what destroys a prion. So, right, we have some ideas, but at this point, something that is sterile is not necessarily prion-free in any way, shape, or form. So sterilization, because the bacterial endospore is the most difficult one to eradicate. Once they are destroyed, everything else is gone. Viruses, bacteria, fungi, etc. Disinfection is something we only do on inanimate objects, tabletops, food consumables. We disinfect. I'm almost done. These are governmental terms. Decontamination and sanitization are set up by a government agency. They have nothing to do with healthcare. Okay? If someone tells you to decontaminate, basically you're disinfecting. If someone tells you to sanitize, you're disinfecting by whatever protocol they're using. The term degerm is stupid. Okay. Antisepsis is something we do on living tissue. You use antiseptics on your skin, on inside when you use a mouthwash. That's it. That's an antiseptic agent. You only use that on living tissue. We'll stop it here and we'll pick this up in earnest. Yes, ma'am.
The review for exam 1 under week 1 does not load.
It didn't load.
No, there's an error.
Oh no.
Didn't for me.
It did not.
No.
What?
Want to see what it looks like? You could probably find it on YouTube. It's on YouTube.
I will try. Okay. Let me see. If not, I'll get it re-recorded. That's not a problem. Yeah, unavailable.
No.
Did you click it? I wonder
Yeah,
if you downloaded it.
I clicked it and it just took me to a link YouTube. It doesn't let you like get a website or
Like the
Okay.
other links don't. Like the other links will take you right to it. They'll start playing, but this one won't.
I wonder if there was a
What does GMT stand for?
Greenwich Mean Time. GMT is like all time kind of comes from time zero when you talk about it. That was interesting. I've never had